Hello and a very warm welcome to everyone joining us for today's Select Science webinar titled Mapping the Epigenome of the Neuroprogenitors in the Embryonic Mouse Forebrain Using Cell Sorting and Single-Cell Transcriptomics to Characterize Interneuron Diversity. My name is Georgina Wynne-Hughes and I'll be moderating today's presentation. I'm delighted to be joined by a guest speaker, Dr. Timothy J. Petros from the Eunice Kennedy Shriver National Institute of Child Health and Human Development. In this webinar, Dr. Petros will discuss his research on how intrinsic genetic programs and environmental signals interact to generate interneuron diversity. Following the presentation, we will have time for a question and answer session. If you do have any questions throughout the webinar, please feel free to submit these by typing into the questions box to the right of your screen. Without further delay, I would like to hand over to our speaker, and I would like to thank them for presenting for us today.
First, I want to thank the organizers for giving me the opportunity to present my lab's work at this webinar. And my name is Tim Petros, and I'm an investigator at NICHD at the NIH in Bethesda, Maryland. And today I'm going to tell you about some of our work where we've been establishing what we call an epigenome atlas of neuroprogenitors in the mouse embryonic forebrain. And this first slide is one of my many failed attempts for a cover image for a journal, but it actually nicely shows the focus of my lab. We study the cell types over here on the left. On the left side is a hemisection of an embryonic mouse brain. And these green cells here are cells that will eventually be GABAergic inhibitory interneurons. They're born in this region of the brain during embryogenesis, and then they go and migrate throughout the brain and populate all brain regions that you can see here on the right side in an adult brain section. And they form a vast array of different connections and are critical in function of nearly every aspect of brain circuitry and brain function. They're the main inhibitory drive in the brain. And so how you generate this incredible diversity of GABAergic cells in the adult forebrain from a relatively small population of cells in embryogenesis is a very exciting and very challenging question to try to understand. And my lab tries to take a multimodal approach to understanding these early cell fate decisions that occur in the embryo in the mouse brain. So as you guys can envision, generating a human brain is quite a complex and long process that takes many, many months, and for it to fully mature, many years postnatally. It starts off as kind of a long tube of cells in the first couple weeks of embryogenesis, and then eventually develops into a brain with approximately 80 plus billion neurons with trillions of synapses. And just to kind of give you a relative comparison, this is the human brain. Everything here is to scale, so you can see the progression of brain size and complexity along the evolutionary chain, both in terms of kind of side views over here on the left and coronal sections through the brains of various species over here on the right. And the organism we study in my lab is the mouse, and you can see that despite its small size in comparison to the human and higher primates, it still is incredibly complex in terms of different cell types, connectivity, morphologies. So it is a very useful model to understand development and organization in the brain of higher vertebrates. Now, my lab utilizes a lot of single-cell sequencing technologies in order to study cell types and cell fates. And so to just give a little bit of background, there's a lot of technical aspects that in the early days was manually picking a single cell to sequence. And over the last 20 years, the technology has advanced incredibly such that you can now sequence millions of cells at the same time. You can do spatial transcriptomics where you put slices on grids and can identify gene expressions within small regions. And there's new techniques being added every month that give scientists the power to really understand cells, transcription, and epigenetic states at the single cell level. Just to give you a feel for this timeline, the first paper for single-cell sequencing was published back in 2009, and they literally published a paper sequencing one cell. So it would be wonderful if we could still do that, but that's not possible now. And you can see that this only goes to 2017, where the advent of 10x genomics has really helped commercialize the ability for labs such as myself to do single-cell sequencing. And now, if you extend this out to 2023, there are many papers that are published that sequence millions of cells in their particular brain region or other organ. So the power of this technique has only grown. In addition to harvesting single cells for sequencing, this requires a lot of computational analysis. And this screenshot is now many months old, but just to give you a feel that as of probably 6 months ago, there was about 1,200 different computational tools to evaluate single-cell sequencing data. And as you can see, this is only increasing exponentially. My guess is it's probably about 1,500 by now. So generating this type of data has become much easier over time, but how one assesses this type of data, this large-scale single-cell data, is an ongoing process and continually being improved with a variety of computational tools for bioinformatic analysis. And this is just one example from the Allen Brain Institute, who has done single-cell sequencing of the adult mouse cortex and also the primate and the human. This image is from the mouse cortex, where every dot represents a cell, and they've done several million cells, and the cells can be classified based on their cell type. So they're all color-coded here based on whether they're an excitatory glutamatergic cell, inhibitory GABAergic cell, oligodendrocyte, astrocyte, and all the different types of cell types. And then they have a nice online tool where all the cells are clustered based on the relationship of the transcriptional profile, and one can go and search the database, type in their favorite gene of interest, and get a readout of which cell types the gene is expressed in the adult mouse brain. So this is just one example of the power of this technique and how it can be available to the public for broad use. Now, to jump back to kind of the cells, I study the interneurons, the GABAergic inhibitory interneurons. This is a nice schematic that kind of highlights the diversity. So this gray cell here is an excitatory cell in the cortex. This receives lots of input and then has output exiting the cortex to go to other brain regions. And you can see all the colored cells here as the vast array of GABAergic inhibitory interneurons that are found in the cortex. And this gives a feel for kind of the different morphologies, complexities, some synapse on distal dendrites, some synapse on the cell body, some only synapse on the axon. And so how a cell interprets all of this different GABAergic input is, again, really challenging and really exciting to try to understand. Now, in the brain, about 80% of the neurons are these excitatory cells here, and about 20% are the GABAergic cells, the colorful cells around here. And as I mentioned, this is an extremely heterogeneous population that has traditionally been classified by neurochemical markers or morphologies or electrophysiological properties, and more recently by their transcriptomes or the gene expression profiles. Now, as I mentioned, these cells are born in the embryo, both in the mouse and in the human. They're born in the ventral forebrain, which is down here, so the bottom part of the brain. At this age in the mouse embryo and the human, it has these eminences, so these bumps that protrude into the lateral ventricle. There's 3 of them, the lateral, the medial, and the caudal ganglionic eminence. And the GABAergic interneurons are born predominantly from the medial ganglionic eminence and the caudal ganglionic eminence. So they're postmitotic, they're born down here. They then migrate up to the cortex and the hippocampus and the striatum. And so they undergo a prolonged refractory migration period to reach their destinations. Now, cells born from the CG and the MGE basically form 2 distinct large populations, non-overlapping populations of GABAergic inhibitory interneurons. Cells born in the MGE express either parvalbumin or somatostatin. Those are fast spiking cells and non-fast spiking cells. whereas interneurons born from the CGE form different cell types expressed by other markers and form different morphologies and connectivities. We know a little bit about some genes that are required for development of interneurons from these two brain regions, but there's a lot we really don't know about these fate decisions that generate these different types of cell populations. And as I mentioned, with the advent of single-cell technologies, people can now harvest single cells from the adult mouse brain and classify cells based solely on their transcriptional profile. Over here in this review, you can see these GABAergic cells here, each labeled with a gene that is enriched in their population. And there's pretty good correlation between transcriptionally defined cell types here and the more traditionally defined cell types by physiology or morphology over here. So there's a good match, but not a perfect match, and there's still work to be done to understand how different transcriptionally defined cell types match up with more traditionally defined interneuron subtypes. Now, one other piece of data that's good to be aware of is how these cells develop during development. So this is kind of a picture of a rodent mouse brain where you have radioglial cells down here. These are the cells that give rise to neurons in the mammalian brain. They can give rise to either direct neurogenesis to postmitotic neurons that then migrate up these radial processes, which eventually become the mature cortex. They can also give rise to what I call basal progenitors, which are cells located here in the subventricular zone, and these cells can still divide and give rise to postmitotic neurons. And why this is relevant is because in the human and other higher vertebrates, you see an incredible expansion of cells of these basal progenitor cells. And this is in large part gives rise to expanded cell numbers, neuronal numbers, and expanded cortex in higher species. So understanding the mechanisms of how you generate this incredibly large population of basal progenitors is quite relevant to humans. And here's just one example of a section through the LG in the human, and what you can see is that the ventricular zone, these radioglial cells are down here, and then you can see this huge subventricular zone that's actually broken up into two regions in the human. These are all cycling cells that are giving rise to postmitotic neurons, in this case GABAergic cells. And so this is just an incredible expansion compared to the mouse, where the VZ and the SVZ are very similar size. Now, my lab recently has been focused on the role that epigenetics or gene regulation plays in formation of interneurons and its role in normal development and disease for a variety of reasons. First of all, there's significant evidence in the literature that dysfunction of GABAergic interneurons is related to both neurological and psychiatric diseases, such as seizure disorders, epilepsy, schizophrenia and autism, bipolar disorder, and a wide variety of diseases. Second, many genes that have been linked to various diseases are enriched in neuroprogenitors, so the young cells present during embryogenesis, as well as in particular in interneuron progenitors, so cells that give rise to the GABAergic interneurons. And additionally, there's growing evidence that epigenetic mechanisms or mechanisms to regulate gene regulation are associated with neuropsychiatric diseases. So many genome-wide association studies or GWAS studies identify SNPs or regions in the DNA associated with various diseases, and many of those regions are non-coding regions. So they could be in enhancers or other regions that regulate gene expression that are not directly in the coding sequence. So this is just a couple of examples. Here's a study from a number of years ago where they were looking at autism-related genes, and they found that many autism-related genes were expressed during embryogenesis in the prenatal weeks, and then they went down in the postnatal weeks. Here's another study from a database that has looked at patients with multiple psychiatric disorders. And if you focus over here, many of the genes associated with these disorders are enriched in the radioglial cells, so those cells in the ventricular zone I mentioned before, and in particular in interneuron progenitors. Now, the advent of studying human cells using organoids has really advanced our ability to understand how genes alter fate decisions and cell mechanisms in human cells. And so here's two studies that have used organoids. And in this study, what they found is that if you look at genes related to a variety of diseases, they are more commonly found in cells giving rise to GABAergic cells compared to glutamatergic cells. And in this study from Paula Arlada's lab, they made stem cells that have mutations similar to human mutations in autism spectrum disorder. And when they did that, if you focus on kind of the blue and purple cells here, in these instances where they made autism-like mutations, they saw an incredible increase in the production of GABAergic cells at the expense of excitatory cells. So there's many other examples in the literature indicating that fate and function of interneurons are related to brain diseases. So kind of an important theme in my lab is that we believe that a better understanding of both genetic and gene regulatory mechanisms that give rise to interneuron fate and maturation is critical for our ability to understand the normal development of these cells and also what might go wrong in disease etiologies. So my lab took an approach to better characterize the state of these cells in the embryo as they're making these critical cell fate decisions early on. And so we wanted to characterize both the RNA expression or the gene expression as well as the gene regulatory state or epigenome state of the cells. So the first thing we did is we performed single cell analysis, both single cell RNA-seq and single cell ATAC-seq, and I'll explain what that is in a minute, in 4 different brain regions that give rise to distinct non-overlapping cell types, the 3 ganglionic eminences, as well as the cortex that gives rise to all excitatory cells in the forebrain. In addition, we characterized the histone modifications in these 4 brain regions so that we could have greater confidence that regions we were looking at were promoters or enhancers and be able to compare and contrast differences in these regions between these aspects of in the DNA. And lastly, we performed a technique called Hi-C and a related technique, Capture-C, that allows us to look at higher order chromatin structure between these brain regions. And we think we've made some interesting insights into how chromatin organization is distinct between these regions and can lead to differential regulation of genes and presumably cell fate. So just to kind of review how our experimental workflow is processed for basically all of these experiments is that we take a mouse embryo, which here is E12.5 stage. We can then open up the brain and hemisect it. And if we look inside the brain, this is a section that's been stained for DLX2, so the brain doesn't actually look brown, but you can see these bumpy structures here. They protrude into the ganglionic eminence, they protrude into the lateral ventricle, so we can open up the brain, see these bumps, and basically microdissect out the MGE, the CG, and the LGE. We then take these preparations and either make single cell or single nuclei dissociations of them, depending on the downstream applications. We sort them, in this case, with the Sony SH800 cell sorter. We then process these cells on the 10x Genomics platform that we have in the lab, which basically is a way to generate these droplets, which contain ideally 1 cell and 1 bead that contains all of the machinery needed to harvest the RNA and make libraries. We do this both for single cell RNA-seq, single cell ATAC-seq, and more recently multiome, which allows us to capture both pieces of information in the same cell. We then send it to our sequencing core, and then what we spend most of our time doing is computational analysis, which, depending on the day, can either be incredibly revealing and insightful or incredibly frustrating as we try to troubleshoot various bioinformatic strategies to perform the analysis. So what is ATAC-seq? So this stands for Assay for Transposase Accessible Chromatin. And basically what it is a strategy that allows us to look for open chromatin regions in the brain. And so a region that's open likely means it's accessible and could indicate a promoter, an active enhancer. And this is basically the way it works. You have a transposase enzyme that can enter the nucleus, and when it identifies a region that's open, like right here, it can bind to this region, it cuts the DNA, and it's been designed to have these adapters that gets inserted. And so basically what you have is pieces of DNA that were accessible or open that now have links, indices on them. Then you can collect this DNA from the cells and amplify and sequence it. And so this is just kind of the schematic that we use. Again, dissect the regions, prep this tissue, and as you sequence it, you basically kind of have this binning window. And in a cell, if you get many reads over a threshold, this is called, this is peak calling. So we would call this a region in the DNA that is accessible in a particular cell. And this is what the plots look like. So this is called a UMAP plot, where each dot represents a cell. And so two cells that are very close to each other have a more similar accessibility profile in their DNA. Two cells that are very far away from each other are quite distinct in their DNA accessibility profile. And what you can see is that cells harvested from the cortex are much more similar to each other than if you compare cells harvested from the cortex versus the MGE, and then LG and CG are much more similar to each other than they are to the GE. To the MGE. There are lots of different ways to look at this. On the left over here, you have putative cell clusters. So these are potentially different cell types or stages of cell division. There's about 25 of them in this data set. This data set contains about 60,000 cells, I believe, from these four regions. We can break down the cells based on whether it's an apical progenitor, so like a radioglial cell, a basal progenitor that I mentioned before, or postmitotic neuron. We can also perform pseudo-time analysis so that we understand the developmental trajectory of these cells. We can also look at the accessibility at specific gene loci. So in this case, this is a nestin gene. The yellow color indicates that the promoter of this gene is accessible in the cells. This gene is expressed in the apical progenitors or radioglial cells. So you can see that the gene is much more open or accessible down here in these young cells. And as the cells become postmitotic neurons, the nestin locus gets closed off. NKX 2.1 is a gene that's restricted to expression in the MGE. This is the MGE population over here. And you can see that the promoter of NKX 2.1 is only accessible in the MGE and it's closed off in these other regions. And so you can do this for a variety of genes of interest. We've also looked at the individual clusters. Again, there's about 25. So if you group the MGE clusters over here, you can see that the NKX 2.1 locus is accessible in these MGE clusters and inaccessible in these other regions. And again, you can perform this type of analysis for any gene that you're interested in. Now, this is kind of a complex figure, but it has a lot of information in it. Basically, we've taken those 25 clusters and color-coded them based on what cell type they are, whether they're an apical, a basal progenitor, or a postmitotic neuron. We've also color-coded them based on the brain region they arise from, cortex, MG, or LG, CG. We've grouped together because they're very similar. And then what we've looked at is the differentially accessible peaks within each cluster. So these regions that are accessible, how similar or different are they between the clusters? With the brighter purple cell being the number of differentially accessible peaks in each, and the size of the circle being the percent overlap. And so what you can see along the diagonal is that that's comparing each cluster to itself. So the size is 100%, large circles, and then the colors are different intensities based on the number of differential peaks. If you focus on the upper left, what you can see is that these are 4 different putative cell clusters arising from the cortex, all apical progenitors. So they're actually somewhat similar to each other. So you see when you compare different clusters within this region, you see a high percentage of similarly differentially accessible peaks between these. So these 4 cell clusters are much more closely related to each other in terms of their accessibility profile compared to any of the other cell types. And so the other thing we've done is that you can look within these differentially accessible peaks in each cluster. You can focus on various transcription factor motifs that are found within these peaks. And you don't have to worry so much about what these genes are. These are all transcription factor binding motifs. And the good news is ones that we expect to be found in the cortical cells are enriched in the cortical processes, are enriched in the cortical clusters, such as NeuroD1, and transcription factors we know are expressed by GABAergic cells are enriched in the GABAergic cell population. This is just one example. So here's EMX1. This transcription factor is enriched in cortical cells, and you can see that the transcription factor binding site or transcription factor motif is predominantly found in accessible regions in the cortical cells and not in the ganglionic eminence cells. So we've done other types of analysis, but one thing that we really wanted to look at is, so that ATAC data just gives us accessibility regions, but we're kind of interested in understanding if there are differences in enhancers and promoters between the different regions. So how this works in the DNA is that you have a promoter over here that's critical for gene expression, and you can have an enhancer that can be located very far away in the DNA that closely interacts with this region to drive gene expression. So we used a Cicero analysis to try to look at co-accessibility. So in every cell, if a region is open at a promoter, in this case the LHX6 gene, we wanted to ask how often is a region upstream or downstream of the LHX6 promoter also open. So the way to read this is that on the middle of this axis here is the promoter for LHX6. You have upstream and downstream of that gene. These big blue purple arches indicate that for every cell where the LHX6 promoter is open, over here in this region of GNA is also accessible. And the higher this peak means the greater percentage of cells, in this case in the MG, that have co-accessibility at the LHX6 promoter, as well as this genomic region down here. So in this particular peak, you can see it's very close to 1. So nearly every LGE cell where the LHX6 promoter is accessible also has a region over here that's accessible, and that could be indicative of a possible enhancer that's regulating expression for LHX6. And again, we've done this type of analysis for the 4 different brain regions for a variety of genes. We've also done it over pseudotime, so looking at changes in co-accessibility profiles in apical progenitors, basal progenitors, and neurons. One quick tangent. So what I've shown so far is this is the single cell ATAC data. As I mentioned, we've also performed single cell RNA-seq data. In this case, instead of looking at accessibility, we're looking at gene expression. We can, in this case, we're highlighting cells taken from the LG, MG, and the CGE. You can see these putative cell clusters. You can look at changes in gene expression profile. You can subset out particular regions. In this case, we were interested in these ventricular cells, these radioglial cells that express high levels of nestin. So we subclustered these out. You can again look at which region they come from, putative cell clusters. You can look at differential gene expression, either from these regions or between these different clusters. And what's critical is that all of this data is great, but you actually want to confirm in the brain that these differential patterns you see in the data are in fact real. So we perform in situ hybridizations on a number of genes that were predicted to have intriguing expression profiles based on the single cell RNA-seq data. And you can see that many genes are kind of enriched in subdomains of various regions, some only found in the LG or the CG. And so we were very happy to see that there's good correlation between our predicted single cell data and the actual gene expression profiles, and we published this paper last year. The reason I bring this up is because we wanted to integrate our genomic accessibility data with our single cell RNA-seq data. We were able to do that computationally. So this is from different cells. So this is transcriptional data and genome accessibility data from different cells, but we could integrate them through some computational work so that they're both occupying the same computational space. That allows us to look at RNA and ATAC together, again, putative cell clusters and pseudotime. And so what we are interested in looking at comparing how enhancers and promoters in RNA change over time. So if we just focus up here, we found a group of genes that are enriched early along pseudotime and then go down as the cells become postmitotic. And so what we found in many of these genes is that the blue is the RNA. You can see that the RNA is expressed higher early on, goes down over time as the cells become postmitotic. And we tended to see a decrease in the promoter, which is the red color, and the enhancer is green here, that the promoter enhancer become closed off after the RNA is downregulated. And we saw this pattern in many genes that we looked at. Conversely, if you look at cells that are enriched later in development, you can see that the promoters or enhancers open up prior to RNA expression. So we can start to look at relationships between gene expression, promoter accessibility, and enhancer accessibility by combining all of this different data. Now, another critical piece of data is, again, all we're looking at is co-accessibility, but can we have greater confidence that the regions we're looking at are actually promoters or enhancers? And so histones are proteins that the DNA enwraps in the nucleus, and these histones can be modified, methylated or acetylated at various residues. And there's a couple critical modifications that give us insight into the state of the chromatin. So one is a trimethylation that occurs at K27. If you see this mark in the DNA, you can be really confident that this region of DNA is repressed. So it's closed off and there can't be transcription happening. Another mark is K4 trimethyl. And this mark is most prominently found on active promoters. So if you see this mark, you can be confident that you're looking at an active promoter where gene regulation, gene expression is occurring. And conversely, a histone 3K27 acetylation is indicative of an active enhancer. So by looking at these modifications within these different brain regions, we can increase our confidence that what we're looking at is an actively transcribed gene, an active promoter, an active enhancer for gene regulation, or a region that's closed off. And so the way we analyze this is through a procedure called cut and tag that was developed by in Steve Heinekoff's lab. And the way this works is that you have a specific antibody that recognizes one of these histone modifications. You then have an enzyme, again, a modified transposase that can bind to this specific antibody. You can activate this enzyme, then causes this transposase to cut the DNA and again, add adapters onto it. So then what you have is DNA that's chewed up and cut at all of these sites that have a histone modification. You can then extract this DNA that has these various adapters, sequence it, and so that you can identify regions where particular histone modifications occur. So this is just an example of the data. Again, we have the single-cell RNA-seq data up top from the 4 different brain regions, single-cell ATAC data, and here I've highlighted the mark that indicates active promoters and the mark that indicates gene repression. So for example, NKX2 is an MGE restricted gene. You can see its expression is only in the MGE, and the accessibility is predominantly found in the MGE. This mark for activation, and again, is strongly enriched in the MGE, and the repressive marks are found in the other 3 brain regions where NKX2.1 is not expressed. I just want to highlight this gene real quick, NR2F2, because it is a CGE enriched gene, as indicated by the RNA, as well as the active and repressive marks. But if you look at the accessibility data, the single-cell ATAC data, it's actually open in all 4 brain regions. And so this is just to highlight that if you only look at one modality, in this case single-cell ATAC, you can actually be misled. This would indicate that the gene might be expressed in all 4 brain regions because it's accessible, but it's clearly not. So this is why having a multimodal approach is critical for truly understanding gene expression profiles and the state of the cell. And so when we look at active enhancers, this mark for active enhancers, K27 acetylation, and combine it with our gene expression data, our ATAC data, and our Cicero accessibility, we can start to make educated guesses for possible enhancers. And I want to point out this one region here. This is a region that has been studied as a possible enhancer for ASCL1. ASCL1 is a gene specifically expressed in the ganglionic eminences. What we found is that although this region is accessible in all the brain regions, it's actually the highest enhancer mark is in cortex here compared to the GEs, where this gene is not expressed. So we'd argue this is actually not a great candidate for an enhancer for ASCL1, since its highest enhancer activity is in a region where it's not expressed. Conversely, just downstream, you see a region down here where there's much greater activation, K27 acetyl marks down here in the GEs compared to the cortex. So we'd argue this is a better candidate for possible enhancer of this ASCL1 than this region that's been previously studied. And so again, we can identify candidate enhancers for various genes known to play roles in specific cell fates. Now, the last piece of data we have is to look at the higher order chromatin structure to kind of understand how this could modify gene expression. And so basically, kind of as I hinted at before, DNA is very organized in the cells. There's a variety of proteins and mechanism that cause the DNA to be segregated from other sections, such that promoters and enhancers can only interact in particular segments of DNA and cannot cross this boundary to interact with other regions. In fact, there's a variety of diseases when these boundaries are perturbed, they can lead to aberrant promoter enhancer interactions and cause problems in gene expression and disease. And so the way to kind of study this is through a technique called Hi-C. Basically, what you do is you kind of glue the DNA in place such that regions adjacent to each other or sort of glued together. You go through a process to cut up the DNA, add in adapters, and eventually what you get is a library full of DNA where two adjacent regions are contacted. You sequence this, and then you can be confident that these regions are in fact touching in the cells. And the way to look at this data, Hi-C data is usually presented like this, and what you want to focus in are these hot spots, these regions of intense red color. And so what that means is that if you follow this kind of down the side of the pyramid, what you're looking at is a region here in the genome that is directly contacted with the region down here. And so this is how you can have confidence that two regions of the DNA, sometimes very far away from each other, are in direct contact in the cells. And again, that could indicate, in this case, maybe a promoter over here is being contacted directly by an enhancer here. So we performed this analysis in the 4 different brain regions. And just to highlight one interesting thing we found, this NKX2.1 gene is critical for fate decisions from the MGE. And what we found is that specifically in the MGE, we see this increase in orange intensity right here, indicating a chromatin interaction. If you kind of go down these 2 regions, that region is absent, or that connection is absent in the LG, the CG, and the cortex. Conversely, there's regions over here where we saw interactions, direct chromatin interactions in the cortex, CG, and LG that seem to be absent in the MG. And then we perform Capture-C to verify this. This region down here in the middle is the NKX2.1 promoter. And what you can see is a region over here where there's a direct contact between this genomic loci here and the promoter of NKX2.1 that's only found in the MG and not found in the other brain regions. Conversely, on the other side of the gene, you see direct interactions from these 2 loci that's present in the LG, CG, and cortex, but that interaction is not present in the MGE. And once you add in the other data we have, single-cell RNA, single-cell ATAC, the various methylation, histone modifications, you can really have kind of a complete story, both at the single-cell level and kind of in the bulk chromatin level, to understand both the RNA and the gene regulatory mechanisms. And one thing that's really interesting in this particular case is this region of interest that has a direct contact with the NKX2.1 promoter is located at this gene called MBIP. And MBIP is actually also strictly expressed in the MG, not expressed in the LG and CG. And so this could be a direct promoter-promoter or enhancer-promoter interaction that's causing regulation or specificity of this gene only in the MG and not the other brain regions. And again, we can look at unique region-specific chromatin interactions for a variety of other genes and loci that provide intriguing candidates for direct genomic interaction that could indicate region-specific promoter-enhancer interactions. Lastly, it's very important for us to make all of this data as accessible as possible. So this paper was published last year in Nature Communications, but we also use the USC Genome Browser to make all of this data publicly available, where anybody can go, type in their favorite gene, and have access to all of this data. So in addition to all of the raw data that's published in the GEO, up here you had the single-cell RNA, single-cell ATAC, our Cicero analysis, all of the histone modifications, the capture-seed data, and down here the high-seed data. And this gene is that we're looking at the NKX 2.1 locus. So what you can kind of make out here's that enrichment, MGE specific enrichment I talked about before. That's again absent in the LGE. So we hope that researchers can go to this website and explore differential expression and gene regulation in these different brain regions in the mouse embryo that are relevant for their research. Okay, so just to kind of summarize, we performed a variety of assays in different brain regions in the embryo that give rise to different subsets of neurons to establish an epigenetic ground truth, so that we now have a good understanding of both at the RNA level and gene regulation level of similarities and differences between regions in the embryonic mouse brain. And I just, again, want to stress that each type of data is only one piece of the puzzle, and we can only truly understand the state of the cells if we try to put many of these pieces together, because you can be sometimes misled if you're only focusing on one type of modality. We believe that our data reveals some novel high confidence enhancers that haven't been previously explored and could provide critical insight into mechanisms regulating specific genes that are critical for cell fate. It does appear that promoters enhancers become accessible prior to mRNA detection, which makes sense, they sort of have to, but they also seem to remain open after the mRNA levels are decreased. And just one example, we highlighted this, we do see distinct brain region specific chromatin architecture at genes that are critical for regulating cell fate, in this case, NKX 2.1. And so now what we're doing in the lab that I don't have time to talk about is we're expanding upon this. Now that we know this ground truth, we're interested in exploring what happens in models of disease where gene regulation mechanisms are perturbed. So we have a variety of mice in the lab where that are either have perturbations in aspects of gene regulation or are related to human diseases, and we're performing some of these assays in these mouse models to understand what goes wrong and what is changed in these mouse models where gene regulation is perturbed, so that we can hopefully better correlate it with disease, with human diseases. So I want to thank everybody in my lab that helped out with this data. My former postdoc, Chris Rhodes, performed most of the data, single-cell ATAC data. Dong-Jin Lee performed much of the single-cell RNA-seq data analysis, and a wide variety of people in the lab helped out, in particular the bioinformatics core of my institute, as well as the sequencing core. Thank you very much, and I'm happy to answer any questions you have.
Thank you, Tim, for that very informative presentation. So it's now time for our question and answer session. So if you do have any questions, please send those in now. You can submit these by typing into the questions box to the right of your screen. So to kick off our Q&A today, my first question here for you is, are there advantages in working with single sorted neurons versus nuclei? Are there any neuronal cell types where working with nuclei is preferable?
Yeah, thanks for the question. So my lab has performed a fair amount of both single cell and single nuclei sequencing, and while there could be slight differences if you're dissociating whole cells versus nuclei, we find that the single nuclei are much easier to work with. You don't have to worry about cell viability, especially if you're performing these assays in adult brains where you have a lot more myelin and debris in the solutions. It's just easier to use FACS sorting or fluorescent markers to harvest single nuclei rather than whole cells. And there's a number of studies that have compared the results between single cell and single nuclei. and in general, the types of cells one can obtain from this data are very, very similar. So the biggest downside of using nuclei is that you lose RNA floating around in the cytoplasm. And so depending on the genes that you care about, maybe genes that are enriched in synapses, you might be losing that RNA more so in a single nuclei compared to single cell. But otherwise, we believe single nuclei is our preferred way to perform single cell sequencing assays.
Great, thank you. My next question here is, are there any best practices that your lab follows to get high yield of single sorted neurons for RNA-seq assays?
Yeah, there's we as did many labs, we struggled with kind of figuring out the best procedures to use to clean up brain tissue and harvest the cells we want. And so we actually published a manuscript last year in STAR Protocols where we detail our preferred protocols to harvest both single cells and single nuclei from embryonic and postnatal brain tissue for a variety of downstream assays, all of them that I talked about in the seminar. And so I'd recommend that people check out that paper in STAR Protocols for anybody that's interested in doing this. One big take-home message is, especially if your sample is dirty, so adult brains, adult spinal cord, we strongly recommend performing FACS sorting to get rid of as much debris as possible. You can do that whether the nuclei are fluorescent, or if they're not fluorescent, you can just do DAPI staining to harvest them. And that really helps clean up your sample for just about any downstream application. So we found that to work very well and be extremely helpful.
Thank you, Tim. What types of epigenetic changes are typically seen in neurodevelopment disorders and diseases, and are these inheritable?
Yeah, so that's a good question. It's very broad and difficult to answer concisely, but it's certainly true that some of them can be inheritable. There's a wide variety of mutations that affect gene regulation. So these would be mutations not in the coding sequence, but possibly in an enhancer region that could perturb the function of that enhancer, or some sort of mutation and a histone residue that can be modified. And in general, what these types of mutations can do is alter the ability for a gene to be regulated either at the proper time or the proper expression levels. And so slight modifications in the gene regulation can, over time, lead to quite severe disease. GWAS studies, so studies looking at mutations in human and relating them to disease, most of the hits or genomic loci of interest in these GWAS studies are actually in non-coding regions. So they're almost certainly doing something involving gene regulation, either mutations in enhancers or mutations that affect the way the higher order chromatin can interact, leading to changes in gene regulation. So that's just kind of and we don't really understand those very well, but we're getting better and better at kind of recognizing them and correlating specific gene loci with regulation of particular diseases. So the general concept of how epigenetic changes regulate genes critical for neurodevelopment is in its very early stages of our ability to understand that, and hopefully in the future provide therapeutic mechanisms for these types of interactions that are disrupted and cause neurodevelopmental disorders.
Great, thank you. And I think we'll leave it there for today. So I'd like to thank you again, Tim, for the interesting presentation and discussion, and a big thank you to everyone joining us online. We hope you found this a worthwhile session. If you do have any further questions, please feel free to email me at editor@selectscience.net, and I will follow up with your questions for our speaker. Remember, you can download related resources in the tab to the left of your screen, and this includes the certificate of attendance. If you'd like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in just a few days' time. Goodbye, and thank you once again for joining us.